I want to say thanks to Paul for being willing to come out on short notice to do a response to this lecture, and also for being willing to give a lecture, as Mark said, and then have me give a response later. I think that sort of charitable dialogue that hopefully we can model will be something that can be an example for others. And I appreciate Paul as a friend, and I'm glad that he's here to do that tonight. So the title I chose was deliberately provocative to try to get you out here tonight so you would hear from me. And I thought, well, we could have a more academic sounding title, but then you might think, oh, what's he actually going to talk about? So I thought, I'd nail my colors to the mast right with the first slide and the title so that you would know what we're on about tonight. So I'm going to present to you tonight on evolutionary creationism, or the idea that God uses evolutionary processes as a means of creation. And we'll work through some of my thoughts on that. And then I'll also talk a bit about intelligent design and why I don't find intelligent design a satisfactory response to evolutionary biology. And of course, that's what Paul will respond to, and then I will have an opportunity to respond to his lecture in a few weeks. So we'll just we'll jump in and we'll see where we get. And then after that exchange at the end, then there'll be opportunity for you to ask questions of both Paul and myself. Okay. So the first point I would like to argue is that science is actually a God-given means for exploring his creation. So we can think of God's special revelation in the Bible, and we can think about natural revelation in the created order. And we can understand science as a means, a God-given means, to use human intellect that he's given to us to explore his creation and understand its workings. Now, I would argue that this is this is an activity that's right in the center of Christian conviction and has been for a very long time. So here's a quote from Abraham Kuyper. Now, I'm not a Calvinist, but I always like to put this in because it's an excellent quote, depending, regardless of what theological tradition you adhere to, where Kuyper makes the strong point that everything in the created order is God's. There's nothing that's not God's. So anything that we can find in the created order or understand through the tools of science, if it's properly understood, then it's something that we can appreciate as part of God's revelation to us. There's also a long tradition in in Christianity and science, where Christians have seen science as a very Christian activity. So here's Francis Bacon, a very central figure in how science gets started in Christian Europe. And he says, "Let no man think or maintain that a man can search too far or be too well studied in the book of God's word or in the book of God's works." So Bacon here is advocating what others have called the two books model, that God has authored two books, the book of nature that we can understand through the tools of science, and he's also authored scripture through its inspiration, and that both that revelation and natural revelation are two books that God has written, and we can understand both of them. Another way to think about this is that God is the author of both special revelation and general revelation. And that inherently, because God is the author of both, there should fundamentally be no conflict between those two forms of revelation. Because again, God is the author of both. Now, humans, on the other hand, have to investigate or appropriate this revelation through human effort. So on the side of special revelation, we employ exegesis or interpretation and hermeneutics, the attempt to understand and interpret what God is saying to us through special revelation. Now, for general revelation, I believe that God has given us the scientific method that we can use to explore the created order. Now, the challenge is that there may well appear to be a conflict between the findings of one and the other. But remember that the assumption, or the baseline assumption, is that God is the author of both, so fundamentally there should not be a conflict. Although our limits for exegesis and hermeneutics and our limits in the scientific method might limit our ability to understand what the actual coherence is if we do see an apparent conflict. So, despite the possibility of apparent conflict between science and scripture, both are sources of God's revelation to us, and Christians are not well served by ignoring or denigrating either. So, I would argue that a true Christian response to God and his revelation has to fully embrace both aspects of his revelation to us. This is, of course, consistent with the Trinity Western Statement on Creation that some of you might be familiar with. It says a biblical view of creation does not constrain legitimate scientific inquiry and research because we accept two sources of information, biblical revelation and natural revelation. Again, underscoring that conviction that God is the author of both. So what I would argue then at this point is to say that well-supported conclusions of science then are a witness to God's revelation. They're a form of natural revelation. They may not be perfect, we may not understand them perfectly, but well-supported science, Christians can look at that and say, this is a form of God's revelation that we have understood through a process that he has given to us. Just as hermeneutics and exegesis can help us understand scriptural revelation, special revelation. So then the question then becomes, well then, what is well-supported science? And this is often a place where Christians disagree. They will say, well, okay, certain things are well-supported, but evolution isn't well-supported, that sort of thing. So then the next part of the talk is just simply walk you through some of the reasons why I think evolution is a well-supported scientific theory. Before we get into that, though, let's just briefly talk about what the word theory means and how it has a different meaning scientifically than it does in colloquial usage. So in science, a theory is an explanatory framework that's withstood repeated experimentation, and it continues to produce hypotheses that make testable predictions. So in colloquial usage, theory is more like a guess or conjecture. This is something that a scientist would say is more closely affiliated with the term hypothesis. So when your friend says, well, that's your theory, what they're really saying is, well, that's your guess. But in science, if you say something is a theory, you're saying it's a well-tested explanatory framework. So if you say something is only a theory, you're actually complimenting it from a scientific viewpoint, if indeed we're using the scientific term for theory instead of the colloquial one. So what I'm going to argue is that despite what you may have been told as a Christian, evolution is a theory in the scientific sense. And I just put this here to ease the pain of that particular revelation. So even goldfish crackers seem to argue about this. So what I'm going to argue tonight is that evolution is a well-tested explanatory framework. It's supported by a large body of experimental evidence. Of course, we're only going to be able to see a very small amount of that evidence tonight just because of time constraints. It makes accurate predictions, and we have not yet falsified it through experimentation. So what a theory is, another way to put this is that theories are never proven in science. We can disprove things, but we can never prove things. What you have in science with theories are converging lines of evidence and explanatory framework that has not yet been falsified. But there's always that provisional idea that someday down the road we may possibly falsify it with new information. Okay. Interestingly enough, evolution is the most foundational theory in biology. This is a very famous quote from a Christian, an Orthodox Christian population geneticist in the 70s. He said, "Nothing in biology makes sense except in the light of evolution." So of course, Darwin was the first individual to propose a coherent model for how evolution might work. And one of the hypotheses that initially got Darwin thinking about evolution is this hypothesis that species that we see in the present day may share common ancestors in the past. So what he proposed, and he proposed that natural selection, differential reproductive success based on variation in the population, he proposed that that might be the actual mechanism which drives evolution over time. So his hypothesis was that populations may become separated genetically in some way, although Darwin didn't have a good idea of how genetics worked. As we would say it now, we would say that genetic changes in those two populations, mutation, recombination, if those populations have now been separated in some way, the differences that are accumulating in those populations are not going to be averaged across the whole. What means it puts them on separate trajectories that may, over time, lead to enough differences that they would not recognize each other as the same species if they came back together. So this was Darwin's hypothesis for how species might form through a process of natural selection, mutation and natural selection. So that was his basic idea. Evolution, obviously, since the time of Darwin, has accumulated a large amount of experimental evidence to support it. And what's really interesting to me as a biologist is evolution continues to produce a large number of hypotheses that can be tested. So the overall framework of evolution is there, but there's details that have not yet been investigated. But it provides this amazing framework for making testable predictions about what you should find in the natural world. One of those predictions is that modern whales should be the descendants of four-limbed terrestrial mammals. Now, I don't have the time to lay out all the evidence for why that hypothesis is a good one. It has to do with comparative anatomy and physiology, what it means to be a mammal, and the probability of all those sets of characteristics cropping up independently in separate species. So if evolution is on the right track, then we would expect whales to be the descendants of four-limbed, land-dwelling organisms. Now, that's pretty crazy when you think about it, because whales are so beautifully adapted to an aquatic environment. And actually, Darwin was ridiculed for this hypothesis. Darwin realized that this was one of the implications of his theory, even back in the 1800s, and he was widely criticized and ridiculed for it, by Christians even, that such an outlandish hypothesis was even possible, and there wasn't evidence for it, they would say. Now, that's a hypothesis. There's many different ways that you can test that hypothesis. One of those hypotheses has to do with what we find in the fossil record. If indeed whales in the present day share common ancestors with four-limbed organisms in the past, then we would expect the fossil record to at least record some of those steps along the way, or more likely relatives of those steps along the way, at least showing us what sort of forms were present at different times in the geological record. Now, in Darwin's day, and actually up until the 1980s or so, a lot of the proposed, or a lot of the expected forms had not been located in the fossil record. And even up until the early 1990s, you'll find individuals, Christians even, who are saying, this is an obvious problem, these forms should be there, but we're not finding them. As it turns out, the place where you really want to do this paleontology happens to be in India and Pakistan and other parts of that area that are great for doing paleontology, but because of political unrest are often difficult to access. So once it was worked out where this had happened, a large number of discoveries quickly followed once paleontologists realized that this was the place where we should be looking for these sorts of organisms. The point that I'll make, without going into too much detail, is that the fossil record, as we now understand it, is replete with all sorts of organisms that blur the distinction between what we consider a present-day whale and what we would consider a proper four-limbed terrestrial tetrapod four-limbed organism. So that prediction that evolution made, that we should find these forms in the fossil record, has been very strongly supported, which means we have not yet rejected this hypothesis based on the evidence that we've seen. Other lines of evidence also contribute to this understanding. So another hypothesis or another prediction that this hypothesis makes is that we might possibly, not guaranteed, but we might observe things in present-day whales that are suggestive of a terrestrial ancestry. One of the places where you might look for something along these lines is in embryology, early whale development. So whales are, the technical term for that is cetacean, so you'll see the term cetacean there. That's just so you can feel smart after going to a biology lecture, right? You can say, well, actually, modern day cetaceans, and people say, wow, they know a lot about science. It's really cool. It's all about looking smart, right? So if you look at cetacean development, in this one example that we'll look at here in a minute, dolphin embryos, what's really interesting is you see some features of their development that is very difficult to explain apart from a terrestrial ancestry. So one of the things that we see obviously in present-day whales is that lack of hind limbs. Whales are mammals. Mammals are supposed to be four-limbed organisms. Whales are kind of the one odd set out. So it suggests that, as we like what we see here in these fossil whale relatives, we would expect at some point that modern day whales have lost their hind limbs at some point in their lineage. What's really interesting though is that modern day whales are in fact tetrapods. They do have four limbs, but they only have four limbs for a very short period of their embryonic development. So just like any other mammal should, early on in whale development, whales form four-limb buds and they form hind-limb buds, just like you would expect them to if they were a tetrapod four-limbed mammal. Now, what's really interesting about this is that later on in embryogenesis, these hind-limb buds are pulled back into the body wall. We've actually done, biologists have done some of the molecular biology of this situation, And it's actually a developmental program. The original developmental program starts running that says make hind limb buds and make forelimb buds. And then later on, a second developmental program kicks in only in the hind limbs that says, no, don't do that. Come on back in. Pull them back in. So you end up with this interesting situation where whales are tetrapods, but only for a very short period of their development. Now that's very difficult to explain away from an evolutionary understanding of their origins, though some have tried. One of the lines of evidence in the present day that's often used to sort of address these questions of who is related to whom is to use DNA sequencing. So as you might expect, we've now looked at the DNA of present-day whales and asked the question whether or not their DNA shows any signs of previously having been adapted to a terrestrial way of life. And the short answer is very much so that we do. So just as two examples, whales have the genetic sequences that other mammals have for air-based olfaction, for smelling based on things that are diffusing in air. It's a large set of proteins that are expressed in the nasal epithelium. They bind onto chemicals in the air, and that binding then sends certain signals to the nervous system that we interpret as smell. Whales have the same genes for that as any terrestrial mammal should have, but the vast majority of them have picked up so many mutations that they're no longer functional. So the genetic scars of those genes are still there, or sorry, the remnants of those genes are still there, but they're scarred with these mutations that indicate to us that they're not functional. Some whales actually don't even have an olfactory organ as far as we can tell anymore. It's just a straight shot from the blowhole to the lungs without any innervation that would indicate a sense of smell. Now, of course, this makes good sense because whales are not dependent, specifically toothed whales, for example, in this example, they're not dependent on using a sense of smell for hunting. What do whales use for hunting instead of a sense of smell? Well, they use echolocation rather than smell. But yet they contain those DNA sequences just like any terrestrial mammal should have. So what we see in this particular example is that we have converging lines of evidence that support the same hypothesis, that whales are the descendants of terrestrial ancestors. And we see it in their DNA, we see it in their comparative anatomy and physiology. That was the first line of evidence that suggested this relationship. We now see it in the fossil record, and we see it in embryology. Okay, you might say, fine, whales evolved, but humans are different, right? Well, the interesting thing about evolutionary biology is that it's not only shown us that all life is related apart from humans, but humans are also a part of this process as well. And again, we don't have a huge amount of time to go through a large amount of evidence, but I'll show you just a few snippets to give you a flavor of what sort of evidence is out there. So maybe I'll put it this way. If we had no idea about evolution before we started sequencing genomes, the fact that we started sequencing genomes and comparing DNA sequences between different species would have forced us to this hypothesis, even if no other evidence had been available. So what I'm going to talk through now are some of the lines of evidence that support the hypothesis that humans and other forms of life are related. Now, it's long been hypothesized that other great apes are our closest relatives. Of course, Darwin was ridiculed for this as well. You've probably seen the caricature of Darwin like an ape. He was ridiculed for it. But we now have the ability to test this using DNA sequencing. If we could go back in time and tell Darwin, hey, guess what? Organisms have what is pretty much like a digital record of their evolutionary past encoded inside them that you can compare with others. He would do backflips if he could. Okay. So when we take modern genomics and apply it to humans and chimpanzees, what's interesting is that the human genome and the chimpanzee genome are very, very highly identical to one another. Now, it depends a bit on how you do the analysis and how you count blocks of mutations instead of single DNA letter changes. But a conservative estimate of how similar these two genomes are to one another is about 95%. That's including blocks of mutations, deletions, and insertions. What's interesting is that we not only have the same genes with highly identical sequences, we also have them in the same spatial pattern. We have them in the same order, pretty much, with a few small changes. Another way to put this is that our genomes are exactly what you would predict as slightly modified versions of what was once a common ancestral genome. So the hypothesis that humans and chimpanzees share a common ancestor has been very, very strongly supported by genome sequencing. This is just a picture to show you what this looks like at the genome level. So on the one side, on the left-hand side of each pair, what you see is the human chromosome, and then it's matched up with the chimpanzee chromosome next to it. And the banding pattern just gives you a visual impression of how similar the two genomes are. This is from the 1980s. This was done a long, long time ago. We now, of course, have sequenced over this at the individual DNA sequence level, so we know that the pattern that you're seeing is consonant with the actual DNA evidence. If you want, in the Q&A, you can ask me about some of the differences. One of the ones, human chromosome 2 is an interesting difference. What we see as one chromosome in humans is present as 2 chromosomes in chimpanzees and other great apes. I don't have time to get into it now. If you're curious, you can ask in the Q&A. Okay, in whales, we made this observation that it's often possible to see the remains of genes that have lost their function once a mutation has happened. I liken this, or use an analogy to describe this. If we have a look at this building, if you came across this as you were walking through a field, you would instantly know a few things about it. You would know that it once had a function, and you would know that its function is no longer what its original function was, simply based on the features that it has. So molecular biologists have this same ability. Genes have certain characteristic features that are necessary for them to do their jobs. Sequences that say, start transcribing here to make a copy, that's an RNA copy, start translating here to make a protein, that sort of thing. So there's a number of structural features that are necessary for a gene to do its job. Now, mutations can remove the function of a gene by inactivating some of those important sequences, but without removing the entire gene, such that you can see that there's a remnant of it there, even though you're confident that it no longer has its original function. Just in the same way you could say, well, this building obviously had a roof, it had a floor. I mean, I suppose it would be possible to say that this was an artificially constructed set, maybe to look like that. But in most cases, if we encountered this, we would say, no, this shows a sign of history. So pseudogenes, they're called pseudogenes for historical reasons. Pseudogene just means false gene. It's a bit of a misnomer. A better way to think about it is defective remnants of a once functional gene. What's interesting is that when we started sequencing the genomes of other organisms, we started noticing, and this is why I say that if we had never come up with the idea of evolution, genomics data would have dragged us to this hypothesis. When we started sequencing genomes of different organisms, we started noticing that many of organisms that were thought to be related based on physiology and whatnot, they also shared mutations in pseudogenes that were identical between different species. So the idea here is that it could be possible, if you did have species that were related to one another, that if you have a mutation happen in a gene that removes its function, if that population of organisms later separates and goes on to form 2 species, then both of those species would inherit that same genetic mutation that was present before they went their separate ways. So these are olfactory receptor genes, the same thing that I was just talking about in whales. Primates have also lost a large number of olfactory receptor genes, humans included. And what we've noticed is that many of these genes, defective genes, have identical mutations in different primates, so humans, chimpanzees, gorillas, orangutans, and so on. The pattern of these genes exactly matches the predicted pattern of ancestry based on other lines of evidence. So maybe I'll just quickly go to the board just to show you sort of how this works. I'll try to stay loud so that the recording can still hear. So what we see in species is we see one of 2 things. We see mutations that are unique to an individual lineage, and we see some mutations that are shared. So these 3 mutations here are identical in both humans and chimpanzees. These 3 are identical in humans, chimps, and gorillas. These 6 are identical in all 4. You should also notice what we don't see. We don't see a situation where we have the exact same mutation in humans and in gorillas, but not also in chimpanzees. If we see the same mutation in humans and orangutans, we can hypothesize with great confidence that we'll also see it in chimpanzees and gorillas, and we do. So these sequences form what's called a nested hierarchy. Okay. Now, what's interesting is, and this actually was very, very useful to scientists because it helps resolve the relationship between closely related species. So there was some discussion before this sort of data came in, whether or not gorillas were more closely related to us than chimpanzees, for example. But this sort of pattern shows us that we share a more recent common ancestor with chimpanzees than we do with gorillas. What's really interesting about this sort of evidence is that it can extend in a lineage for a very, very long period of time. So here's one example that my students have all seen a million times, so they know what's coming. This example of a gene called vitellogenin. Vitellogenins are large proteins that egg-laying organisms use to put nutrition into the egg yolk before the eggshell is deposited. So they're a nice source of amino acids and sugars and lipids all in one. They're made in the liver of the mother, and then they're transferred through the bloodstream to the egg yolk before the eggshell is deposited. Because obviously once the eggshell is deposited, the mother can no longer contribute. Now, placental mammals, on the other hand, don't need these because we maintain a placental bloodstream connection with our embryos all the way through their development. Now, again, I don't have time to lay out all the evidence for you, but the hypothesis is that mammals are an innovation within a group of egg-laying organisms. So another way to put that is the evolutionary prediction is that mammals are descended from egg-laying ancestors. You might be familiar with, there's two lineages of egg-laying mammals that are left on the planet, the platypus and the echidna. So those are egg-laying mammals, and they still use vitellogenins, but marsupial mammals and placental mammals no longer do because they maintain a placental connection of one form or another. So what this research group was interested in was simply asking the question, could it be the case that there are the remains of these genes in placental mammal genomes, even though placental mammals have not likely shared a common ancestor with egg-laying organisms for a very, very long period of time. So the way they did this is they looked at an organism that has functional vitellogenin genes. So they happened to choose chicken because chicken is a good egg-laying organism. Its genome is sequenced. We know about where its genes are. Chickens have 3 vitellogenin genes, and you can see them there. 2 of them sit side by side in the genome, and 1 is in a different genomic location. What this research group did was to say, what sort of sequences are close by to the vitellogenin genes in chickens? So that you can see the genes there, ELTD1 and SXX2, SSX2IP, that sort of thing. They simply noted what genes were next door. What's interesting is in both cases, the same genes are sitting in the human genome in the same spatial arrangement. So what they simply asked the question was, if indeed humans and chickens in this case do share a common ancestor, we may well see remains of these vitellogenin genes in the human genome at this point in their sequences. And when they did the sequencing, they found that indeed that was the case. So what these black bars indicate is where there are sequence correspondences between human and chicken chromosomes. And you'll notice that it spans both regions on either side. And you'll notice that some of the shared sequences actually do sit inside where the vitellogenin sequences are in chicken. So we can tell when we're looking at a sequence whether or not it's a coding sequence to make a protein or not. And we see fragmentary remains of the protein coding sequences for these 3 genes in the human genome, exactly where you would predict them to be if indeed we do share a common ancestor with chickens. And just for fun, they also looked at opossums because these sequences should be in all mammals. And opossums are an example of a marsupial mammal, and they found the exact same arrangement there. So opossums and every other placental mammal and marsupial that they looked at also have fragmentary remains of these genes in their genomes. Now, despite the many mutations, as you might expect, because it's been a very, very long time since these genes were functional in our lineage. So they've accumulated many mutations, but what's interesting is that many of these mutations are shared between all placental mammals that we know about. So I'll just go back to the board for a second here. So for example, when you look here, everything below the top line is a placental mammal of some kind. You'll notice some of the sequences, or you'll notice in this case there's a DNA letter that's missing, and it's missing in every placental mammal. Here's another one where it's missing, and again, it's missing in every placental mammal. This mutation here, or these 3 letters here, this is an amino acid, or this is a code to the amino acid translation machinery to say, oh, you're at the end of the protein, stop making a protein. Except this is really, really early on in the protein. This should not be here at all. And we see that that's also shared by all sorts of placental mammals. So the idea is that when we see these shared mutations, it's most likely the case that these mutations cropped up before these different mammals separated from one another. So that, again, as I say, is just a small snippet of the sort of evolutionary evidence that we can find. I could talk to you all night about all sorts of interesting lines of evidence, but those are the ones that I chose to show you. Let's change gears now a little bit and ask, okay, it seems like there's good evidence for evolution, but what about intelligent design? As Christians, we believe in design, right? So what does that mean? That sort of thing. All Christians believe that God is the creator and that he is the designer and that he intentionally created what we see. But intelligent design, when it's got the capital I and the capital D on it, is a technical term for something a little bit different. And Dr. Brown, of course, will get into this a lot more in his talk in a few weeks, and also in the response. Intelligent design is, again, it's a technical term. So let's listen to some of the leaders in the intelligent design movement to hear in their own words what they think intelligent design is. So this is Michael Behe. He's a biochemist at Lehigh University in Pennsylvania. And he says, intelligent design is a scientific theory that proposes that some aspects of life are best explained as the result of design, and that the strong appearance of design in life is real and not just apparent. So what is design, he says? This is from one of his books. He says, design is simply the purposeful arrangement of parts. When we perceive that parts have been arranged to fulfill a purpose, that's when we infer design. So in this sense, and again, Paul can correct me if I get any of this wrong later, because Paul's an expert in this and I'm not. Intelligent design is a proposal that certain features of what we observe in the living world are actually the result of, they would say, design. I would say that's a very similar idea as to special creation, that these are not something that came through an evolutionary process, but came through the deliberate actions of a designer who's instantiates design at a certain point. One of the arguments that Behe makes in support of intelligent design is something that he calls irreducible complexity. So Behe says that irreducibly complex systems indicate design since they are beyond the ability of normal evolutionary processes to generate. So what does Behe mean by irreducibly complex? He says, by irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. An irreducibly complex system cannot be produced directly, that is, by continuously improving the initial function, which continues to work by the same mechanism, by slight successive modifications of a precursor system, because any precursor to an irreducibly complex system that is missing a part is by definition non-functional. So typically examples of irreducible complexity are complex biochemical apparatus that we find in the cell that do specific jobs, and they're composed of numerous interacting proteins that do a specific function. So the argument Behe is making here is when we see those sorts of parts and that they're arranged to do a job, we infer that there's purposeful design behind them. And he would say they're irreducibly complex in the sense that when you take one of the parts away, the system stops functioning. So what Behe would say is, therefore, that's evidence that parts could not have been added to the system in the first place through an evolutionary process, because the function is not present until all of the parts are in place. Now, I'm also summarizing 2 books by Behe in a few slides. So again, come back in the Q&A, and I'm sure Paul will have some interesting things to say about irreducible complexity as well. Behe's argument rests on the claim that mutations that are needed to form these irreducibly complex systems are too improbable, because he would argue multiple mutations are needed in order to assemble these protein complexes and get them to work together. So Behe looks at these complexes and says regular evolutionary processes can't assemble these because you need all the parts to be together in order to have a function. So Behe and I have had a fair bit of back and forth on this over the years. Here's an example of one of the things that I sort of posited to him and he had a reply to. As a fruit fly geneticist in my professional life, I often like looking in the fly literature, and I came across this paper and I thought, oh, this is an interesting example that perhaps says something about Behe's argument. So again, remember, the core of his argument rests on this assertion that you need multiple mutations, simultaneous mutations, in order to generate new irreducibly complex systems. In fruit flies, this one at the bottom, melanogaster, is the standard laboratory research fruit fly. But they're a member of a large group of species, and we've actually sequenced the genome of about 12 of them now. What's interesting is when you have those sorts of DNA sequences of related organisms, you can scroll through, as it were, and look for things that are different between the two. And in this case, melanogaster happens to have 2 copies of a particular gene, whereas the other members of this group of relatives only have 1. So in simulans, Shellya, and melanogaster, we see this gene called eclair. Fly geneticists are notorious for naming fly genes with all sorts of wacky names. I'm sorry. The mouse geneticists get very sort of put off by this, right? A mouse geneticist has nice, logical names. And a fly geneticist, I mean, you know David Suzuki, right? He was a fly geneticist, and he was very much in that same spirit of, you know, very carefree and easygoing, and yeah, we name them all sorts of crazy things. I mean, who came up with P24.2? Sounds more like a mouse geneticist started being a fly geneticist there. But eclair, yeah, that's the kind of thing we name our genes. Anyway, so melanogaster has this gene called eclair, and right next door to it in the genome is a gene that's almost identical to it that got named P24.2. What's interesting about these genes is that they're virtually identical at the amino acid level. Don't worry, there won't be a test at the end to test your memory on this. This is what's called single letter amino acid code. So genes are made up, they're coded for by a DNA sequence, and then that sequence is used to make amino acids in a specific sequence, and the amino acids form the protein that does the actual job. What's interesting about these, if you look at these, we can represent amino acids with just single letters, and that's what you're looking at here. The point is that they're virtually identical. There's only 5 differences. One of the differences is here, and 4 of them clump together at the end of the protein. Now, we know what this category of proteins does. They're used in tracking or transporting other proteins around the cell. So what likely has gone on here is that we've had a duplication of the same gene side by side in the one genome in melanogaster. And then the duplicate ended up picking up a new function because of those changes. It's now transporting something else as opposed to what it was originally transporting. And we've actually done the experiments in flies where we can knock out eclair, as an example, and to see if P24.2 can pick up the job. And they can't. They're no longer redundant, and they're both necessary. If you take either of them away, the fly is in trouble. So I proposed this to Behe and said, it looks like we've had a series of mutations that have converted one protein to another function, and that function seems to involve protein-protein interactions. What do you think about this? Kind of thing. And I was making the case that this would seem to undercut your assertion that simultaneous mutations are needed. Well, Behe replied, and he said, okay, there are 5 specific amino acid differences between eclair and P24.2. Professor Venema seems not to comprehend that they may not have arisen by random mutation. Once one gets beyond 1 or 2 random mutations, one can't assume that multiple further mutations arose by chance. In other words, as far as anyone knows, those 5-point mutations may have required guidance or design in their appearance. So here's the important point. What Behe is saying is, once you get beyond 1 or 2 random mutations, you may well be into the area of design, where things actually have to intentionally be done, where a designer actually has to do the mutations, and natural selection can't do the job. Now, I actually thought that was reasonably surprising. I thought that Behe would be actually fine with this as an example because of the other lines of evidence that support the idea that it's a duplicate, they're sitting side by side in the genome, there's a very small number of changes between them. but it was informative. Okay, so then the point is that short of a time machine, I can't resolve this to Dr. Behe's satisfaction. I would actually need to track whether or not these mutations actually happened all at once, or whether or not they happened, as I'm proposing they did, one by one, along a trajectory to forming a new protein-protein interaction. So short of rerunning the last couple million years of fruit fly evolution, it's just not possible. Now, the same sort of question, however, can be addressed in systems that are more tractable, easier systems to work with. So here's an example that I'll just run you through to illustrate how this sort of thing can be done when we actually do have the ability to watch a population over time and see what sort of mutations take place in it. So this is an experiment that was set up to test the ability of a certain bacteriophage. This is a virus for bacteria. So we have our viruses. Bacteria also have their viruses as well. So this is a virus that infects E. coli, the standard sort of gut bacterium that humans are familiar with. So what this research was interested in was this research group, they were interested in the question of what sort of changes are possible within this virus in terms of how it interacts with its host cell. So lambda phage, this bacteriophage, this bacteria virus, is a DNA virus, so it's called a class 1 virus, and it's specific to its host, E. coli. So the way that this nasty little virus infects E. coli is it uses one of its viral proteins to stick onto a protein in the bacterial membrane, and that's the start of the infection cycle. The protein that lambda uses is called LAMB. Again, this is not a fruit fly geneticist naming names, but anyway, it's a much more respectable type of scientist. So this virus uses this protein in the bacterial membrane as a way to start its infectious cycle. These things are called porins. This is what it actually physically looks like somewhat when you have a crystal structure, what it looks like. It's kind of like a tube that sits in the membrane. The virus sticks onto that tube and actually puts its genetic material through the tube and into the bacteria. So, and here's what the bacterial membrane looks like. E. coli have two membranes. So LAMB is this thing here. The virus sticks onto it, puts its genome through, and then it actually goes through other things to get inside. But this LAMB protein is absolutely essential for lambda to infect bacteria. So this group was interested in this setup, and here's what they did to investigate it. They were curious if they took LAMB away, if the virus would sort of figure out an evolutionary workaround to taking away that receptor. Now, if you take it all away at once, if there's no LAMB left in the bacteria, then the virus can't replicate and it dies. So they cleverly set up a system where only a very, very few bacteria in their mix would have the appropriate receptor. So there's very rare bacteria that allow just a few viruses to replicate. But if anything crops up that allows them to use something else on the bacterial surface, then that variant would have a huge evolutionary advantage because it would be able to infect bacteria that the other viruses wouldn't be able to. So what's really interesting about this experiment is when they did this, they found that the virus easily found another way to get into the bacterial cells. And it did so by using another porin, another protein that is physically similar to LAMB, but not related to LAMB at the amino acid level. So it's physically similar, but it's not identical. Now, here's the interesting thing. The workaround actually requires 5 mutations in order for it to work. These viruses have to have at least 5. Some of them had more. And if they didn't have those 5, they couldn't use this other receptor. So the research group was curious about this. They thought, okay, it's not possible that these 5 mutations could be cropping up simultaneously, so what's going on here? That's too improbable. So what's actually going on here? What's interesting is that in every case, the 5 mutations that were needed could be found as single mutations in individual viruses. And those single mutations actually allowed the virus to stick onto the original LAMB protein more tightly. And it just so happened that once the virus accumulated 5 of those mutations, all of a sudden, boom, it could use this other receptor, this other protein that it couldn't use before. So in other words, those five mutations did not need to be simultaneous. You could have them crop up sequentially, one by one, until suddenly this new function that wasn't there before appears. So now we could say that OMPF, this new porin that the virus is using, we could say that it's now one of several parts that have an arrangement for a purpose. When that OMPF is present, the virus can use it now as a means of getting into E. coli. Another thing to think about is that if you take OMPF away and you take LAM-B away at the same time, now the virus isn't able to infect the bacteria at all. So this actually forms a system that meets Behe's criteria for an irreducibly complex system. We have a set of proteins that are arranged to allow the virus entry into the cell. If we take one of those proteins away, then the system doesn't work anymore. So we see parts arranged for a purpose, and we also see that the parts are now necessary. Except in this particular case, we actually have concrete evidence that the system transitioned through an intermediate state where we had two optional proteins. You could use either LAM-B or OMPF before you transitioned over to a state where now OMPF is required. Okay. So that's one argument from intelligent design. There are a few other arguments. Stephen Meyer is another example of an intelligent design proponent. Some of you might have been here a little while ago when we did the streaming of the online debate that had Stephen Meyer in it. One of the arguments that Stephen Meyer makes for intelligent design is that there is no other means of producing biological information other than a designer. So if we see biological information in a system, then we can infer that it was designed. So here's one quote from one of his books, Signature in the Cell, in 2009. He says, "Since the case for intelligent design as the best explanation for the origin of biological information necessary to build the novel forms of life depends, in part, upon the claim that functional, information-rich genes and proteins cannot be explained by random mutation and selection, this design hypothesis implies that selection and mutation will not suffice to produce genetic information." So all that to say is, if you're claiming that intelligence is the only source of biological information, then if you see biological information, then you can infer that design was responsible for it. But in order to make that claim, you have to be able to support the idea that other processes can't produce what you're talking about. So if a natural process could be found that could produce biological information, then that would render this argument problematic. Okay, so the question then becomes, are information-rich genes and proteins possible for evolution through mutation and natural selection to produce? If so, then merely observing biological information will not be sufficient for us to infer that it was designed in that sense. Okay, I'll just walk you through a few examples of evidence from evolutionary biology that supports the hypothesis that evolution is capable of producing substantial amounts of new biological information. So here's an interesting study that was done a number of years ago. This is a long, long time ago in evolution, back in the Cambrian or perhaps the Precambrian, where the origins of vertebrates, so things with skulls and backbones, originate. There's long been this hypothesis among biologists that vertebrates may have had a couple of rounds of what's called whole genome duplication, that the genomes that we and all vertebrates have are actually doubled or perhaps even doubled copies of an ancestral genome, where you have basically a failure of cell division, where you have, instead of dividing chromosomes, you end up with both chromosomes in one cell, and then that actually isn't a problem at that particular point. It's a problem for many organisms, but we actually know of examples in the present day where that sort of thing can happen, and it's not fatal to the organism. So there was long this hypothesis that this may have occurred right at the beginning of vertebrate evolution. One of the reasons why we sort of thought this was the case is because of the numbers of genes that we see in vertebrates. Vertebrates seem to have a number of copies of certain types, categories of genes, whereas some organisms will only have one set of those genes. Vertebrates will have 4 sets. So the hypothesis was kind of, well, maybe genome doubling was the result, the process that produced this. The challenge with that is it's hard to differentiate that hypothesis with the idea that maybe you just had small chromosome duplications of like regions of chromosomes that just happened to build up a couple of sets of genes as opposed to doubling everything in the genome at once. So it wasn't until we actually were able to do whole genome sequencing of a large number of organisms before we could really test this hypothesis. So if you had rounds of genome duplication, this is the pattern that you would expect to see in present-day organisms. And I'll just go to the board again to walk you through it. So at the top, if you have sort of the ancestral state, whatever it was, and the different colored boxes just represent genes along a chromosome, the first doubling event would just make 2 copies of everything, but it would keep them in the same spatial pattern. Then over time, what would likely happen is that some of those copies would be lost. At the point of the duplication, everything's redundant. You've got 2 copies of everything, and you don't really need 2 copies of everything because just prior to the duplication event, you were doing just fine with just 1 copy of everything. So over time, it's likely that some of them would be lost. Some of them might be maintained as duplicates if indeed this becomes a preferred situation. Then if this happened again, this would now be the baseline step or state for the second duplication event. But again, the overall pattern of things in a specific order would be maintained. And then again, from that point on, you would expect that some would be lost and others would be retained. So you end up with a range of things. You could see cases where you have 4 copies, some cases where you just have 1, others 2, others 3. But the key to this whole thing is the pattern, the physical relationships of where these genes are relative to one another. Now, the question then becomes, well, which genes should we look at? We want to make sure that we're looking specifically at the genes that would have actually been present at the time these supposed doubling events were proposed to have taken place. And the way you do that is you can look at vertebrates and their closest relatives as a way to figure out how that goes. So I'll go back to the board here for a second. So tunicates, these are things called sea squirts. They're the closest relatives to vertebrates that aren't vertebrates. So any gene that a tunicate has that vertebrates like tetrapods like us and fish have, those genes would most likely be present in the last common ancestral population of all of those species. If we see all of those genes in present-day species. Now, anything that we see just in fish and tetrapods might have cropped up later on and wasn't present at the time when these hypothetical duplications took place. So what this group was interested in was they went through the sea squirt genome and compared it to the human and the fish genome, and they found all the genes that were in common between those 3 groups. And they said, okay, these are the genes that would have hypothetically been present in the last common ancestor. When they specifically look at those genes, that subset in the human genome, this is what they see on the left there. So I'll just walk you over to it so you can see what's going on. Hopefully the tape can still hear me too. So when you look at this, you'll notice that there's sort of these arc patterns. So the lines connect simply where the copies are. So if there's a gene here, this just shows you where the gene is on another chromosome. So these are chromosomes within the human genome. Human chromosome 10, human chromosome 2, and so on. And what you see is that there's this large spatial pattern of where the copies are. They're all in a certain arrangement on one chromosome, and all the copies are in the same arrangement on another chromosome. Just as you would predict if in fact these were the result of whole genome duplication events. Now, when you look at the subset of genes that is only unique to vertebrates but not present in sea squirts, we see a different pattern. And that's this pattern here. So here what we see is often that the copies sit side by side on the same chromosome. So that's much more suggestive of local duplication events as opposed to this whole genome duplication event. So all that to say is that we have this genomic pattern that suggests that we did go through our lineage deep in the past, went through these whole genome duplication type events. But now here's the kicker. Those different genes in present-day vertebrates don't have the same identical function. They've picked up different functions over time. So they're copies, they're similar to one another, but those individual genes have picked up new functions. So that's an example of new information showing up over time. Okay. Here's another example, and this is one that I mentioned at the panel discussion that we had with the Meyer, Lamoureux, and Krauss debate. Sometimes you can actually have new information show up seemingly from nowhere, as it were. So here's an interesting example, and this is one that I think Paul and I talked a bit about last time. This idea of nylonase. This is an enzyme that degrades a man-made chemical called nylon. This is a bacterial gene. Nylon was first synthesized in the 1930s, and by the 1970s, scientists had actually identified bacteria that were able to digest nylon and use it as a carbon source. They were feeding on it. And this is a man-made chemical, so it was quite surprising to them that these bacteria were now able to live off of a chemical that hadn't existed in their environment prior to, 40 years prior to them discovering this. And here's actually how this gene came about. So this is an enzyme which cuts up nylon into smaller fragments that the bacteria can then use as a carbon source to feed on. So the mutation that gave us this, it was a single mutation, a single DNA letter that was inserted into another gene. And this single DNA letter at the same time did two things. It produced a stop codon in the previous gene, so it gave the signal to this gene to say, okay, stop making protein here, which of course completely ruins the function of that gene, whatever it was. But at the very same time, that same mutation also made a start, saying, oh, start making something here. And that start actually has a run of many, many amino acids, over 300, and that structure, that protein, which kind of came out of that sequence, has a very rudimentary ability to digest nylon, to act as a nylonase, to cut up nylon. Later on, what happens in this organism is that this gene duplicates, so now you have two copies of it. One copy stays pretty much as it was, and the other copy then undergoes a number of mutations that make it a very, very efficient nylonase, such that the bacteria that have it now can use nylon as a sole food source. It's the only thing that they need to live on. So here's the interesting thing. That's new information. It's a new function. It's come out of nowhere, as it were. Well, not really. The DNA letters were there, but the amino acid sequence is completely different from what we saw in the previous example, the previous gene. The interesting thing is that Meyer says that this can't happen. If you remember, for those of you that are familiar with Meyer's argument, you'll know that he says that functional sequences are too rare in the world, of the available possibilities. We should never see something like this happen. He says that functional sequences are on the order of 1 in 10 to the 77th power, the probability of randomly finding a sequence that's functional. And that's actually only for a sequence that's 150 amino acids long. This sequence is over 300. So Meyer from that infers that the functional information that you see in proteins could only be the result of intentional design. But we have examples here where we can look at and say, well, actually, we have good evidence that this happened through mutation and natural selection, and yet produced something that was functional, folded up properly, and did a specific job. We can also look at the human and chimpanzee and gorilla genome for this sort of thing too. So we talked about how fruit flies, you can look at different fruit fly genomes and find new stuff. Some scientists are very interested in new human stuff. Is there anything that's specific to the human lineage that isn't present in other great apes? And what you can see in this case, here's one example. We can find some brand new genes in the human genome that are made into proteins that are not found in any other great ape. But what's interesting is that when you look at the sequences in the same spot in the genomes of those other organisms, those other great apes, the sequences are there and they're very, very close to becoming a gene. There's only 1 or 2 mutations that separate them, those sequences, from what we see as a protein coding gene in the human genome. So they're not coming out of nowhere. They're coming out of a very, very similar sequence in the human genome, which strongly suggests that mutation and natural selection was actually what produced them, as opposed to design in that sort of special creation sort of sense. Okay, Meyer would say, okay, you're talking about genetic information when there's already a genetic information system present. What about the ultimate origin of this? Where did this information system originally come from? So Meyer says, in order to explain the origin of the genetic code, scientists need to explain the origin of the precise set of correspondences between specific nucleotide triplets in DNA, or codons on messenger RNA, and specific amino acids carried by transfer RNA. Molecular biologists have failed to find any significant chemical interaction between the codons on mRNA or the anticodons on the tRNA and the amino acids on the acceptor arm of tRNA to which the codons correspond. So what Meyer is arguing, this is all about how the original DNA genetic code originated. And Meyer is saying we can be very certain that this is the result of intelligent design because there are not mechanical, chemical means by which it could have been produced. So he says, this means that the forces of chemical attraction between amino acids and these groups of bases do not explain the correspondences that constitute the genetic code. The code is physically and chemically arbitrary. All possible codes are equally likely, none is favored chemically. So when Meyer and I reviewed this book, Signature in the Cell, and you can find that online if you Google, Venema, Meyer, and it's in a journal called Perspectives on Science and Christian Faith. Meyer and I have a back and forth with a couple of articles. And one of the things that Meyer makes the case for is that he says, okay, Dennis, even if information might happen through duplication events within a genome, what you haven't yet explained is where the original DNA system came from in the first place, back at the origin of life. And we're quite certain that that would have taken design in order to implement. So Meyer is absolutely right that scientists do not yet have a causal explanation for where that system comes from. It's an absolutely challenging, very difficult system. It's very ancient. It goes right back to the origin of life on the planet. So as you might expect, it's actually very challenging to study. So Meyer is absolutely correct that scientists have not yet given a fully causal explanation for how this might have originated. But what's interesting is this argument that Meyer is making here, he's saying, and one of the reasons why we're quite certain design must have been involved is because the code is chemically arbitrary. So DNA is read off in those letter groups of 3, like we were just looking at on the previous slide. And Meyer is saying any amino acid could have, in principle, been assigned to any group of 3 DNA letters. It's an arbitrary code, just like a designer would make. So the question then becomes, okay, is the DNA code chemically arbitrary? Here's just a diagram to get you an idea of what it is that we're talking about. So the way that the DNA is turned into a protein is the nucleotide sequence is read off in these groups of 3 letters. And they're read off by these little adapter molecules. So these are small nucleic acids that physically bring the amino acid in. And they line up on the nucleic acid copy of the gene in order to bring in this amino acid in the right order. So what Meyer is saying is any amino acid, those little things up at the top there, could in principle have been hooked up to any single one of these 3 DNA letter sets. And he says, there's no evidence for any chemical correspondences. Now, what was really interesting is in the same year that Meyer published his book, a paper came out that actually refuted that particular argument that Meyer made. So here it is. So this is a paper that came out in 2009 called RNA Amino Acid Binding, a Stereochemical Era for the Genetic Code. And what this group found was that, in fact, when you look at some amino acids, they actually do physically stick on to the 3-letter code of their tRNA, their anticodon. So some amino acids, those little things up at the top, actually physically stick onto the 3 nucleotide letters that their adapter molecule uses. So what Meyer is saying is a chemically arbitrary code, actually for a subset of it, actually looks like physical chemical interactions could actually be part of how this system came to be in the first place. Now, that doesn't provide a full explanation for where the code came from, but it gives me pause to think, this is an area where research is still progressing. Something that Meyer claims shouldn't be there is actually there, and it makes me wonder if in the future we may find more evidence that suggests that Meyer may not be making a correct inference. All that to say is that design arguments have a long history within Christianity. And what we see with Meyer and Behe and others is sort of just the modern equivalent of a long tradition within Christian circles. So you probably weren't reading new publications back in the late 1600s. This is a book that was published by a fellow named John Edwards, not Jonathan Edwards, the Puritan theologian, but an individual named John Edwards in England in 1696. It's called A Demonstration of the Existence and Providence of God from the Contemplation of the Visible Structure of the Greater and Lesser World. And that's not the whole title. You can see the rest of it there. My editor nowadays would never let me get away with a title that lengthy. Okay. Here's an argument that Edwards makes in this book. He says he's actually talking about certain things that he sees in the natural world that are not explainable based on known scientific understandings at the time. And one of the things that he latches onto is that suns and stars are these flaming balls of gas. Well, they didn't know what they were at the time. And he says, it's amazing. They burn and they burn and they burn and they never burn out. And there was no physical process known at the time that could sustain combustion at that level for that length of time. So he says, and if it be asked, whence is that fuel for these vast fires which continually burn? Whence is it that they are not spent and exhausted? How are these flames fed? And he says, none can resolve these questions but the Almighty Creator, who bestowed on them their being, who made them great and wonderful, that in them we might read his existence, his power, and his providence. Now, the interesting thing here is that what Edwards doesn't know about is he doesn't know about the process of nuclear fusion, obviously, because nuclear fusion is a long, long ways off. So there's no reasonable explanation for how this works in the present day. And what Edwards does is he says, from this we can see that God exists and that he cares for us, because there's no scientific explanation. Now, I would argue that the process of fusion is part of God's providence, but I wouldn't use it in the same way that Edwards is using it. So in modern language, Edwards is actually making a design inference. He's saying that there's no known causal explanation for the longevity of solar combustion. He's saying God can maintain solar combustion, perhaps via miracles or adding fuel in some way. And he's saying since there's no natural explanation, we can infer from solar combustion that God exists, and that this is an inference to the best explanation. Now, Edwards also is very, very clear on his purpose for writing this. He says, if I cannot by this attempt convince atheists, yet I hope that I shall do something toward preventing the spreading of that pernicious infection of which they are the authors of. Or if my hopes fail me as to this, yet I will not despair of confirming and strengthening such who are really persuaded of the doctrine here treated of. So the reason why he's bringing these arguments to the fore is to strengthen the faith of Christians against atheist attack. So here's where I sort of land on this. I ask myself this question: are design arguments that rely on a lack of evidence a good idea for Christians to employ? And the tricky thing is that we don't often know in a given day and time how the science might play out in the future, right? Have we really hit an impasse? Or could future work potentially undermine what we see as a sure thing, a sure argument in the present day? And one of the things in that back and forth I have with Meyer, if you look it up in the articles, is I press him on this point and I say, this isn't a good idea to sort of pin your hopes on this certain understanding because it may well be undermined in the future. And I press him on the fact that origin of life research is still progressing and it hasn't reached an impasse. Here's a quote from Dietrich Bonhoeffer that I like to use. This is written, it's in a book called Letters and Papers from Prison, when he's imprisoned. How it is when he is imprisoned in Nazi Germany that he's reading books on science, I do not know, but the respect I have for the man is great. He says this, he says, Weizsäcker's book, The Worldview of Physics, is still keeping me very busy. It has again brought home to me quite clearly how wrong it is to use God as a stopgap for the incompleteness of our knowledge. If in fact the frontiers of knowledge are being pushed back, and that is bound to be the case, then God is being pushed back with them and is therefore continually in retreat. We are to find God in what we know, not in what we don't know. God wants us to realize his presence, not in unsolved problems, but in those that are solved. All right, so in conclusion, this is sort of where I come down on this. It's possible to look at this whole conversation as an idea of evolution versus design. That evolution is somehow at odds with a providential God accomplishing his purposes in creation. Based on the evidence that I see for evolution, and also somewhat on what I see as or how I don't find intelligent design arguments convincing, personally, I look at it a little differently. I don't think of it as evolution versus design. I think of it as this, that perhaps evolution actually is the design. Maybe what we're actually looking at when we look at the natural order and see the revelation that God has provided to us through the scientific method as a form of natural revelation, maybe instead of thinking of it in terms of somehow against God or not design, perhaps another way to think about it is to think that evolution actually is a form of design, that this is an intentional process that God has put into the biosphere to allow biodiversity to develop that will be well integrated with itself and will interact properly as a unified whole. Now, that's a slightly different philosophical way of thinking about design, but it's one that I find more profitable personally. And with that, I think I will close and invite Paul to come up and do his response. Thank you very much. Now, this is going to be me in the hot seat a couple of weeks from now, so it's hard when you hear a lecturer to suddenly respond. So if you want to see me in the same position, come back in a couple of weeks.
But I will say Dennis is very good and very cool and very good at responding. So yeah, I appreciate Dennis's concern and arguments. We both were actually, he's currently in a Templeton Foundation seminar series on science and Christianity, where he gets to go to Oxford for a couple of years. I got to go to that for 3 years.
Yeah, you got 3, I only get 2.
I know, I got a good deal. Back in 2002 to 2005. So I've heard many of these arguments before. And yeah, we're responding. Sometimes, I don't know if you ever get the feeling sometimes when you see a debate like this or something, it feels like people are talking past each other a little bit and not really addressing one another. And so I'm going to do that. I'm going to do that right now. But I think it's in part because there are some different frameworks, some different ways of looking at things. And I think in intelligent design, for me, it starts with quite a different framework. One of the things that I've always been interested in is when there are disagreements and so on, okay, where are the points where we diverge? Okay, so for example, Dennis started off with a nice discussion about science and that there's God's natural revelation. We know, of course, as a Christian, we would say there's God's special revelation in Scripture. But then there's His natural revelation. We want to take that seriously. So we would agree on that. Okay, so we both do science. And again, in one of those ironies, I don't know, maybe I'm the only professor on campus that's actually been involved in publishing original research on testing evolutionary hypotheses because of my work with chemical ecology. So I've been around the hypotheses, I've been around to see the predictions, and I've been around to see sometimes the failures of those predictions, and to see ideas that I thought might be more correct actually verified. And perhaps I haven't made up my talk yet. This has all been fairly quickly put together. And so, but maybe I can even present a little bit of some work that I was involved in. So one of the things, I was surprised by the talk actually a little bit, in the sense that there was so much devoted to refuting intelligent design. But that's fine. Because I was going to spend a little bit of time, perhaps, well, maybe stating some concerns that I have about evolution. I think, although perhaps it wasn't stated explicitly, a lot of things were stated by Dennis implicitly. But I think actually, again, if I go back to where's those points of divergence, where do we start disagreeing, and what are the downstream ramifications of that? And I find that very often it goes right way back to some very fundamental assumptions that we both make about nature and about God. And yeah, one of those things I was going to say in this talk, I tend to be empirical, so one of the differences is experimental evidence, but Dennis actually presented some this lecture. I think there can be alternative, perhaps, explanations for those, but that's actually good, so I'm glad to see that we're dealing with some empirical data. But I think this fundamental assumption, there's a big difference, and I hope to explain this in more detail when I have time to give a fuller talk. But maybe I'm just gonna, can I walk over? I want to draw something on the board. Yeah, so one of the things I just find is there's a fundamental difference in what we allow in our explanatory framework. And so Dennis mentioned the God of the gaps idea. That's a very common conception and criticism of intelligent design. I actually don't think that that's what the issue is. I don't think it's God of the gaps. So I tend to think this is just an illustration. I just thought of this a little bit earlier. And so I'm just going to attempt to draw this out. Let's imagine we have an explanatory framework. And that explanatory framework is what we will call natural causes, or what we can call physical causality. The evolutionary framework essentially makes the assumption that everything can be explained in terms of natural causes, physical causality. Because of that, then, is the assumption that everything can be explained that way. So if we find something that doesn't fit with that kind of explanation, it's simply attributed to a lack of our understanding. So that's where the God of the gaps comes in. It's considered an argument from ignorance. So we just don't know what the natural explanation is yet. But I think for us, and so what happens is you kind of have this idea that we have bits of knowledge, and that knowledge is on a line in this plane, because everything in terms of evolutionary theory, of course, has to be connected. We start with an origin of life event, and all of life is explained by the same kinds of mechanisms. So historically, all life throughout the history of life, all the information is generated, etc. The origin of the coding information systems and so on, everything can be explained in these terms. And so what we imagine, and one of the benefits of being at the Templeton thing was I actually got to talk this out with people like Ernie McMullen and present this view and have him say, yes, that's right, that kind of thing. So what happens is we imagine that there's this line, we have these data points, they have to fit on this line, and then gradually over time we keep adding more data points, and eventually we'll fill this line in. But what if it's not that way? What if there's actually another kind of cause that's out there? Well, I think there is. And so we might draw something like this. Let's see if my artistic skills aren't, super good. Okay, so we have a plane, and there's an intersection point here, let's say. So this is behind the plane, and this would be in front of the plane. Okay, so I'm trying to kind of do a three-dimensional thing. Does that work at all? Okay. What if there's another kind of explanation? And we call this intelligent agent explanation. In other words, what if intelligent agency is a real thing? And what if intelligent agents have real effects? Well, then we actually have now, instead of this kind of planar explanation, we could have a sort of three-dimensional explanation across these two different planes. And instead of creating an image that's a line, we might have something that's more like a sphere or something in three-dimensional space. And so when we get a data point here that actually doesn't seem to fit on the line, then we check that. And we say, well, does this data point actually fit more in line with this plane, or does it fit more in line with the physical causality plane? And one of the things that intelligent design does is it gives us the option of actually considering both kinds of explanations. We aren't committed to making one kind of explanation. And I would say that this particular commitment is not necessitated by science. There's nothing about the empirical method that demands it. There's nothing about the data that demands it. There should be some precaution taken because, as Dennis said, sometimes people can wrongly attribute things to perhaps an intelligent agent cause that shouldn't be. A particular example he gave, I think one cautionary note is you don't want to attribute operational causes to that. So orbiting planets, we don't want to have God moving the planets around the sun. Those are operational. But when we talk about origination of things, the fact is that we all know that the origin of things, I just got this cell phone. Welcome to the 21st century. I've never had one, and I still don't have everything. I mean, I just got it. But it's a wonderful thing. But we all know we could explain the operation of the circuits in here in terms of physics and chemistry. There are certain materials, gates, electrons moving, so on and so forth. But if we wanted to explain the origin of this phone, or in fact the origin of almost anything in this room, including your shirt, or your socks, or your pen, or this camera, or this board, or the building that we're in, or the seats that you're sitting in, or your socks or your underwear, none of these would have explanations that can be reduced completely to physical causality. The real explanation for them, in fact, includes intelligent agency. So the question then becomes, what kind of things do agents do? What kind of things do the processes and properties of matter do? Can we look at those, compare those, and then begin to evaluate, when we look at life, what kind of traits do we actually see? What kind of traits do we actually find? And for me, when we begin to apply any criteria you want regarding design, what we find in life is that it's chock full of all the features of design. So what do I do with that? Well, for me, that says something that I need to take that into consideration, and I'm going to actually take both into consideration, and I'm going to try to evaluate that as best I can and look for features of things that are consistent with design, features that are consistent with physical causality. So a crystal has a certain kind of order, salt crystal has a certain kind of order, snowflake has a certain kind of order. We know that physical processes can produce certain kinds of order, but we also know that intelligent processes produce certain kinds of order, and often that order has some very, I would suggest, distinct properties that separate it out from strictly physical causal properties. And when we look at life again, we find that. I mean, that's what we find. So, so then we have to begin this process of trying to evaluate how we put all the pieces together. And so for me as an ID person, that's what I try to do. And anyway, so I think I was supposed to only have like 10 minutes or something. So I could say more, but if you want to hear more, of course, come back in 2 weeks. And at this point, I think, I'm not sure what the format is exactly, but I think, anyway, I just wanted to present something. You know, I'm not going to try to address all of Dennis's things, and there's a lot of circumstantial evidence that I think is quite convincing in many respects. But I also, I mean, what got me interested in part of this was actually But it seemed to me there were real problems in explanation, and I think that's how many of us in ID get started. But as we begin to think through things more, we begin to actually create a whole different way of looking at creation, of looking at the world. And so I don't think it's a simple God of the gaps kind of thing or any such thing.
Michael Behe has a quote which acknowledges as an intelligent design scientist and believer that he must attribute not only the beauty of creation and the wonder of creation and the glory and complexity and all of this, but he must also lay at the feet of the creator such things as malaria. He says if we have an intelligent design working through the development of life, then we have this theological, to me it's a theological issue as well as a scientific question, that we now have God responsible for all kinds of the horrors of creation, where an evolutionist would say, well, God left some random possibilities in his creation, and these are the things that happen, which seems to maybe be a better theology. What you have a response to that?
Yeah, that's a good question, and it is a common objection. So, bad design, evil design. How do we handle what's called natural evil in the creation? When you begin to think about design and how it could operate and how it could be implemented, there are many different levels. So, for example, I tend to break design down into a group like what I call background design, which is design in the physics. I think it's real. I think it's there. And we do get snowflakes, and we do have, many Goldilocks phenomena and so on. I think there's design there, fine-tuning and so forth. But then what I call foreground design, which is when an agent, for example, can make matter into conformations it just doesn't go to on its own. It doesn't mean there's any violation of natural law. There's nothing about the existence of my cell phone that violates natural law. It's just that natural law alone doesn't explain it. So that's just one example. There are also different ways design could be implemented. So you could have front-loading to some extent of design. There could be all different kinds of variation. You can have design on top of design. So if there is design in the physics, every object that we design ourselves is design on top of design. So it's a kind of thing that way. Then you can have non-design on top of design as well. An example I've used is if some of our students maybe come from Ontario, they have a 69 Toyota, Corolla or something that's half rusted out, barely makes it out here and burns oil, and everybody piles in it and trashes it because it doesn't matter because it's highly degraded design. And I think that kind of thing is possible. So you can have non-design on top of design. So I don't know fully how to explain that. One of the things I think also is where I would agree on one theological point, there are probably a few we would agree on, is that sometimes our perception of natural evil may not be God's perception. So if a shark has teeth to eat other organisms, is that evil? Well, I think we would both perhaps suggest maybe that's not evil in the same way that a moral decision by a human would be. So I don't know fully how to answer that question, but I don't think God has to design every single point. And in fact, I think there's real evolution that operates. I happen to think it operates in a very different way than what Dennis thinks. He would suggest, I believe, that it's a highly generative process. And I have had a hypothesis that's quite different than that. But I still think changes can happen.
In the back there. Maybe just wait until Mark points the recorder at you.
In the recent debate by University of Toronto, Stephen Meyer was saying that evolution is random, and as a result of that, some people were commenting, I think Richard Dawkins as well, saying that's not random because of natural selection. My understanding is that natural selection can only select for something that has already been produced. So the real driver of evolution would be random mutation. And natural selection kicks in after random mutation has generated something. So how does Richard Dawkins, or perhaps if you agree with that, how is evolution not driven by random processes with the addition of natural selection?
Maybe I'll speak to that. Paul can speak to it if he wants as well. Natural or evolution as a whole is a non-random process because it has that non-random component within it of natural selection. So you're absolutely right. You have variation in a population that natural selection acts on. But as a whole, even though evolution has a random component to it, I would argue overall that it's a very non-random process because of the selection component that makes it very non-random. So one way that we can see this is through something called convergent evolution, where we have separate lineages of organisms that nonetheless have very, very similar evolutionary trajectories at a surface level. So, for example, the marine reptiles that were present at the time of the dinosaurs, ichthyosaurs and plesiosaurs and things like that, they have that same streamlined body shape that we see in whales and dolphins and other marine organisms, even though they did not inherit those features from their last common ancestral population. So in that case, what we have is we have evolution. Random mutations are part of that process, but you also have convergence, natural selection, shaping those trajectories in very repeated ways. So overall, I would say evolution is not a random process in the sense that it's, anything goes. It's very much constrained by the physical properties of the biosphere, and as a result, it's very reproducible depending on the circumstances. If you want to comment on that.
Just to add a little bit, I think random is itself an idea that I think, could be explored a lot. I think with intelligent design, I don't know quite if this is the best description, but I think a lot of intelligent design people would prefer to say it's an unguided process. Evolution is, as presented in the mainstream theory, is an unguided process. There appear to be random elements to it, and that's where the information generation, in my opinion, becomes problematic because they are unguided. But there are constraints on what the outcomes can be, and those constraints involve natural selection. I happen to think natural selection is often not all that effective. In some cases, it's very effective.
Maybe just another quick comment. Randomness has a whole slew of meanings, some of which are philosophical. And when an evolutionary biologist says that they're talking about random mutation, what they're saying is that those mutations are not correlated with the needs of the organism. So they're random with respect to function. But it goes beyond science to say that they are somehow philosophically random in some sense, or that it's unguided or unsupervised or unintended. Those are philosophical claims that a scientist can't properly make. We have very good evidence that when mutations crop up in organisms, that they're not correlated with the needs of the organism in that specific environment. So in that sense, we would say biologically that they're random with respect to function. But that's it.
I have a question. I don't know whether...
No, go ahead. Mark's just coming with the
I don't know
thing.
if my question can be explained in this lecture. My question is, we talk about organisms which is alive, it's a living thing. Then I'm thinking about non-living things, about rocks, water, sky, and oxygen and gas. And organisms, they have atoms, they have protons, they have neutrons, they are composed of carbon and other elements. However, rocks, non-living things, are also composed of things like that, elements. Then why organisms can change, but But I mean, at the atomic level or at the cellular level or at the more level, but why non-living things they don't change? I mean, they are still rock. I know there is erosion, there is changes like, let's say, melting ice, but I mean, the changes is so different between living things and non-living things.
Maybe one thing I would say is that students that have had me in lectures before know that when you get down to sort of the nitty-gritty details of how life works, there's actually a blurry line between what scientists are willing to call alive and non-life. So things like viruses would be something that most people would know about. Are they alive? Well, they certainly have some of the features of living things, but they lack others. And then there's actually even less lifelike stuff that is similar to viruses. So one of the reasons why I suspect that life may have what we would call a natural origin is because of that observation that the line between what we see as living and non-living in the present is a bit blurry. Now, maybe to respond to something that Paul said earlier, as a scientist, I am actually perfectly open to alternative explanations as a Christian. If I thought that there really was good evidence for something that was not explainable by natural mechanisms, I don't have any prior commitment to say, yeah, I'm only going to go with natural stuff because anything else is just getting God's foot in the door. Like as a Christian, I have no problem with that whatsoever. I just haven't seen anything yet that I find that I'm confident enough in to say, yeah, here we really have found something that we can use to sort of show those atheists that we really have something. And I'm somewhat reluctant for the process in general, as you probably gathered, just because I think that it hasn't had a really good track record in Christianity over the last couple hundred years. Now, that's not to say that maybe in the future something really might be found, and I'm open to that. I just personally haven't seen anything yet that I'm confident in. On the other hand, I'm perfectly okay with other people trying and to see what they can find. The only concern I might have is that I don't really want to get into a situation where young people in our churches have to feel that they have to reject evolutionary biology because to be a Christian you have to reject it or something along those lines. So one of the things I had for my subtitle today, why I think you should accept it too, even if you don't find it fully convincing on your own, I'm hoping that Christians going forward will at least be willing to accept other Christians who feel that it's okay, so that our young people don't feel like they have to make a choice either way. But I fully support the idea that people can investigate and propose other hypotheses. When I've tried to interact with Stephen Meyer and Michael Behe, I've always done my best to try to keep it with the science and not say, well, you're just trying to, sneak God in the back door. Well, it's the same God. We're talking about the same God. So, of course, I support that. But I'm curious, I just have my doubts about the method that they're using.
So here's a question then, Dennis, because we've had some of these conversations before, and it seems to me that you have in the past restricted all explanations to physical causal, but I'm glad if you're open to other things at this point. But what would constitute then evidence for design freedom? Could anything?
Well, I guess in a sense, I think that all natural stuff is part of God's design.
Yeah,
in
the
that
background
sense.
design.
Yeah, so I guess maybe you're asking what would qualify as foreground design? I guess what I would want to see is a situation where the more and more we find out about something, the wider and wider the gap or the sort of blank spot on your chart seems to get. The concern that I have is that it seems to me, and I know that you might disagree, and that's okay, disagreements are okay, it's just that the examples that seem to be given by the intelligent design movement, the more and more we learn about them, it seems like the gaps get smaller, not bigger. So at that point I kind of go, I don't know if this is something that's going to last. So as a result, I'm not confident that this is something that I should build an apologetic on. And the other thing is that oftentimes when I dig into the details, it actually seems like there's misunderstanding underneath the details. So like Behe, for example, with his understanding of population genetics, I look at it and I go, yeah, you say that this is how it has to happen, but then I think, no, that's not how it has to happen. So I guess what would be interesting to me would be something where the harder we look at it, the wider the gap gets, as opposed to what appears to me that the gaps get smaller.
Okay, so that's one criteria, widening gaps. Actually, there are widening gaps. But, I mean, is there anything else? Is there any feature? One of the claims that we make is that design is recognizable. And I've got it in my vest. Anyway, maybe I can make this point in my talk. But, so what features might there be? Let me just give you a couple examples of yours. I think the origin of life gap actually is getting wider in terms of the first life. There was just a recent paper out on the minimal life of a cell, and the gap between prokaryotes and eukaryotes, for example. But those things, in my mind, actually suggest they're not specific fingerprints of design, as you apparently interpret them, but rather points where there might be discontinuities in the seamless chain of evolution, which then, of course, is more consistent with, or possibly consistent with the design hypothesis, and not consistent with the evolutionary one. So it's a little more disconfirming for evolution than perhaps demonstrating design. But are there any actual fingerprints of design? I mean, this is one of the things that we can point to in designed objects, and so.
So what sort of things would you consider, or I don't know if you want to get into?
Well, I'm asking you.
Oh, what I would consider?
Because one of the problems I run into is that it's like there is no evidence that actually counts for design. And so, I mean, if design is a real possibility, there has to be some kind of evidence that can actually count. So that's the kind of thing that I would be interested in.
Okay. Well, I think what I was saying, it would be what I would see as a gap that gets wider, where the more we know about something, the less it would be explicable based on what we know of as natural mechanisms. But even then I would be cautious, absolutely. Because I guess we don't really see, maybe we don't see design in the same way. And maybe in your talk, maybe you'll define exactly what, like, sort of what you see as the hallmarks of design. The thing is like, as you know, design proponents often refer to human artifacts as examples of design and say, like, these things have features from which we can infer that they have, a design history behind them. The thing is with life is it has analogies to living or to human artifacts, but it also has properties that are very unlike human artifacts. Right? So I would be less confident in the design of this chair, for example, if I knew that there were populations of chairs out there that reproduced and had genetic variation and possibly were subject to natural selection. Then when I look at that thing, I might pause and say, yeah, it sure looks like somebody went and made that, but I also know that there might be other explanations, so I would pause at that point.
So if I'm understanding you correctly, you still haven't actually said what criteria would count, but you are saying that even if those criteria were there, you would probably discount them because you believe that there could be natural explanations. Is that correct?
Well, what I would say is that what we observe as natural explanations and what we might account as supernatural, that all of those are within the providence of God. So it wouldn't bother me either way if the chair was a human artifact or if it was the result of what we might call a natural process. Because I don't separate design out as something that's, I don't say it's designed because it's not explicable with natural mechanisms. But I also think of natural as part of the providence of what God has set up. So in that sense, we probably do agree on sort of like the background design thing. I think that the cosmos is engineered by God to have the properties that it does. I just happen to think that evolution and perhaps life with a natural origin might be nested within that potentiality that God put into the cosmos from the beginning.
Yeah, and I think that's a great hypothesis. I just don't think it works. But and I think there's another hypothesis available that's better. I don't know how long we want to continue, because
We'll have the chance in 2 weeks' time to
that's true.
sit here and
That's true too.
do it all over again too. Maybe we should just see if any of these patient folks if there's any lingering questions. Yeah, Yanni.
I'm just wondering how, there's so many theories in science, and as a Christian scientist, how does that theory jive with God's word and with his character? Like, especially if we look at evolution and we look at that process, and then all of a sudden there were people, where did we get a soul? And how does that jive with God's character? If before humankind had a soul, all those pre-kinds, so to say, died, and there was death and destruction. And that, how does that jive with God's character and with his word?
Great questions, thanks. Obviously, in a limited presentation, there's not time to get into all of those things. And actually, as a non-theologian, I'm actually not the best person to talk about it. Who I would recommend on that front is a fellow and a friend and a colleague who's been up here a number of times and lectured here, and that's John Walton from Wheaton. If you go online and Google for a series of talks that he gave called Genesis Through Ancient Eyes, or Origins, Genesis Through Ancient Eyes, he lays out, as a Hebrew scholar, how he sees Genesis speaking not to material creation, but rather to God ordering the functions of the cosmos such that they benefit and are set up for humans. So John would actually argue that we don't have an account of material origins in the Bible. So again, like, the lecture is like an hour and a half long, so that's a start. But maybe what I would say, just now putting my amateur theologian hat on, is it seems that God is very comfortable with process and history to me. And I think we even see that with the story of Israel, that God reveals himself to Israel as his chosen people, and then there's this long winding history before we arrive at Christ. You know, it's sometimes been asked, well, why wasn't Christ crucified, the day after Adam fell? Why that long history? So it seems to me that God doesn't mind process and history, and that he's okay with working his processes and his providence into that history. So that's like a small answer. How does it fit in with death? How does it fit in with suffering? Those types of things, those are all good questions. I mean, one advantage of young earth creationism is that it has a very easy answer for theodicy, right? It has a very, very easy answer for where evil comes from, right? Everything was perfect, Adam sins, and then everything's bad after that. I mean, there are some theodicy questions there, why are individuals why are we all affected with Adam's sin and all that sort of thing. I think in some sense we would all happily be young earth creationists on that theodicy front, except for the fact that the overwhelming evidence that we see in nature doesn't align with that particular interpretation. So based on that, because we have this commitment to looking at natural revelation as a form of God's revelation, and I'm comfortable looking at that and saying, maybe I'll have some questions on how this all fits together theologically. So it's not a great answer, but it's a start anyway. Maybe listen to John, and then we can have coffee at some point and see what you think.
Yeah, I think it's a great question too, and as you know, because we talked in the faculty lounge, and I'm quite content with an old earth, so on that, some of those points we might be a little closer in agreement, but I think the question is a really good one on the nature and character of God. One of the problems that I have with evolutionary creation and theistic evolution is not just the science, but is that very issue. It seems to me we also have to take very seriously the revelation that we have and the nature and character of God, who speaks things into the creation. And God said, and it was so. and also that God is revealed in Jesus Christ. So Jesus is the Word by whom and through whom all things were created. Jesus is the one who speaks to the wind and the waves, and creation responds. So there is a direct interaction with God and creation. I believe that is a legitimate thing to consider, and something that I actually believe that happens. And one of the problems with theistic evolution is that it doesn't just make a statement about the natural world, but about the way God interacts with it. And I think it oversteps its bounds when it claims, essentially, that everything must be explained within the natural paradigm.
I'm not sure evolutionary creationists say that everything has to be explained within a natural paradigm, but I understand the concern.
Yeah, I'm just interested in your point about, let's say, almost like in terms of God's character, because God's unchanging, is the same. And I think the Bible gives a lot of basis for believing that there's a symmetry between God as creator and then God in salvation history, revelation history, bringing about the new creation. And much of the New Testament Christology is actually based on that symmetry of Christ, the creator of the cosmos, and then the incarnate Christ, the creator of a new creation. And having, reading the thing that way, one of the problems that I think this two-dimensional view of evolution would bring about is, okay, yes, you're right that the Old Testament certainly shows that God's a God of process, and he works through history. And yet the Old Testament really shows that God does intervene, input revelational information, and the very concept of revelation, as it's expounded in Scripture by the Apostle Paul and so on, is that indeed it is, kata theon, not kata anthropon, that means according to God, not according to man. And there is the gospel, the mystery is something that the evolution of the human race could never produce. It's not something that could emerge naturally. And so the view that you would get through revelation history is that God indeed not only providentially directs the whole thing, but guides the whole thing by inputting information into the history and moving it forward. And in that sense, at least on that side, you don't have really the analogy or the symmetry with evolution unless you go down the radical liberal theology route and say it's all mythology and try to explain everything simply in terms of the evolution of human thought. And you virtually explain it all away rather than explain it. So in terms of the character of God, if you see it's the same God with the same way of handling history and bringing about creation in that symmetry, I think that Genesis would indicate that indeed the world we have is a physical world with God layering information into it, and therefore the material world of Genesis 1 and 2 itself, unlike the pagan views of the Babylonian and New Age, do not have, like matter does not have the creative potential to organize itself up into these hierarchies that brings about finally the creation of man in God's image. And that's very symmetrical with the view of God in salvation history. So what would you say to that?
Well, again, I probably refer you back to John Walton because I'm not an Old Testament scholar. John's thesis, and I find it persuasive, is that Genesis 1 and 2 is not an account of material origins, and that we, as Western scientifically minded individuals, when we look at the text, when we see material origin there, that we're actually reading our own culture and our own concerns into the text in ways that would not have been a concern or even on the radar of the original recipients of those narratives. So John's laid this out in a couple of books. The first one's called Lost World of Genesis 1, and the second one that came out a little while ago is called The Lost World of Adam and Eve. And the argument that he makes there is that the word create is not talking about creation, creatio ex nihilo at that point. Now we hear that and we think, well, of course that's what that means. Create means God speaking and creating. Now, John fully affirms creation ex nihilo, but he says that that's not a doctrine that you can find in Genesis 1 and 2. What he sees in Genesis 1 and 2 is an account of God ordering creation in a functional way. One of the analogies that John likes to use is to say the difference between a house story and a home story. So if you came to my house and you asked, tell me how your house, how is it that you came to have this home here? You would find it odd if I started talking to you about the material construction of the house and when the sheathing was brought and when the roof was put on and that sort of thing. What you might be looking for instead would be like, how is it that we came to live here? How is it that this house became our home? We would talk about how perhaps we purchased it and the items that we brought into it to make it into a home. So what John is arguing is that what we have in Genesis 1 and 2 is the home story and not the physical material creation sort of house story. So in that sense, what John is arguing is that if we don't have a material narrative of material origins in Scripture, then it comes down to that it didn't seem to matter to God to actually provide that material story for us, and that God was pleased to allow us to work out that story through the scientific method. I don't know, Paul, do you want to jump in?
Yeah, I actually agree with your position, at least in terms of the interaction. And I think the story is bigger than Genesis 1. I actually, I've read Walton's stuff. He makes some good points in there, but I find the overall argument unconvincing. I think there are just better ways to do it, particularly when you take the whole of Scripture and try to develop. I'm not a theologian either, but, we're in this debate, and you learn quite a bit of theology just along the way, picking it up. It seems to me a theology of creation involves a lot more than just Genesis 1, and it seems to me that your presentation of that idea as this process with layers, God interacting with creation, that's much better to what I would think.
Can I just say one thing about John Walton? I have read his book on the lost world of Genesis. I would put forth this challenge that he might actually be more modern, reading the modern point of view of the Genesis, than the conservative Christian, simply because, that is the modern way of thinking, to dichotomize between religion, the spiritual, and the scientific. And the second thing I would say to him is, I do agree that Genesis 1 should not be taken as a scientific document. And I think that Job, God's discourse with Job, shows that, the mysteries of creation are there, and God hasn't disclosed them to us, and so I'm not going to, go that way. But the other side is that in setting up the home, it is the home in which the people of Israel, the Christians, live. And you got to remember that Genesis 1 has a worldview dimension to it. And it definitely is before it's John Walton, God sort of trying to design the universe after the model of the tabernacle. There's a tremendous protest, right, in the first 3 verses against the whole pagan cosmology in which all matter was seen as loaded with spirit, power, and nature, the evolution of God. You see, one of the things you have to face is that all ancient pagan civilizations actually had a cosmology, which in principle was evolutionary. And that is that the forces of nature were the gods. When the unity exploded, they were diversified in the gods, and then that internal evolution from the cosmic egg or unity brought about this world of diversity. And when you read Genesis 1 in that theater, it's very clear that there is a cosmological statement being made, and that is that matter and the world is not divine and does not have a self-creating power. And then, of course, that sets up the home for the children of Israel in the history of Revelation. And that is that God does not leave nature to simply evolve itself into the incarnation. But indeed, God set up the world in such a way that nature itself does not have its self-creating potential, and therefore it's open to God. And he's the one that set it up for him to interact in those ways throughout history.
I guess, I mean, there's a lot there to respond to, and John's capable of defending himself, to be sure. Maybe one thing I would say, though, is that it's not ever a Christian position of any stripe to say that matter somehow has properties of its own that can do things on their own apart from God, right? So if you're hearing the Christian evolutionary creationism position as saying matter is somehow autonomous on its own apart from God doing things and God is not a part of that, that's not a Christian position. That would be at best sort of a deistic kind of approach to things. So, but I agree with you that there are statements being made in Genesis that are saying, no, the pagan mythologies that you've heard, those are wrong. God, Yahweh is the creator. He has put this all together, right? And the whole narrative of Genesis 1 and 2 is that God is ordering this cosmos. So what John would be arguing is, yeah, God is the author of that, and he's ordering it, and any natural processes that we happen to investigate through science would fit into that framework, not that nature would somehow have this autonomous, separate from God kind of quality to it. I agree, though, that, evolutionary creation does cause, deep conversations to be had about theology, absolutely. What I sometimes think about is the flip side, though. If evolution is wrong, if evolution actually didn't happen, then why is it that we have the evidence that we do? And that actually, for me, creates an interesting image of God. Like, either, all of the world's scientists, save a very, 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 very small minority, are absolutely off base. And I've looked at this evidence myself with my qualifications, and I find it convincing. But if that's really not the way things happen, then I have some very challenging questions of theodicy there. It's like, why is it, or not theodicy, but just the idea of why would this sort of pattern be in nature if in fact it's completely not the way it actually happened? So that, then I would have those sorts of concerns.
Simple answers read God's answer to Job. And there's an interesting one, that God has certain delight in, as it says in, I guess, Proverbs, it's the glory of God to hide a thing, and it's the glory of the king to search it out. And one way or another, the universe is full of mysteries. And God probably likes to bring us precisely to where he brought Job, to the wow factor, and to say, you know what, I think I spoke of things wonderful to me. Well, all I'm saying is, I think whatever angle you come at it, you are going to come up against the inexplicable. And, there's two things that philosophically worry and theologically worry me about evolution. One of them being, let's face it, that they are pursuing an atheistic type of explanation. In other words, they're just bent on an explanation of things in which God does not factor into the explanation.
What I would just to respond to that, most scientists don't care about those questions at all.
Yeah, I know.
Yeah, most scientists don't think of their work as atheistic.
Yeah, I suppose it just.
Yeah, they just don't. They're concerned about getting their next grant and explaining the natural world, and they really don't care about whether or not it's philosophically one way or another. But
one other feature, and I'll go quick because I don't want to take over and make a third lecture, is Genesis 1, by bringing in the Sabbath, very clearly makes a statement that in many ways time is divided into two. Creation time is over. And after the Sabbath, it would be, no doubt, with adaptability and a lot of life, because life has to be adaptable. But nonetheless, after the Sabbath, mankind, new races of mankind don't evolve, let's say, or don't get created. But rather, it almost seems to say there is a kind of a time of the world coming into being, and then the time of the world being into being, which would be time of history. Whereas the theory of evolution, of course, knows the Sabbath. And that actually, raises a lot of interesting issues for Christians, and for theology, for ethics. And I think it should raise some very interesting issues for the theory of evolution. Why we don't see races of apes continuing to kind of evolve upward.
Oh, sure we do. And actually, I mean, evolution is continuously taking place. But to get to your point about the idea that, you know, at the Sabbath of creation week, that was the point at which all creation stopped. I mean, they pressed Jesus on that in the New Testament. And he said, hey, my Father's been working to the present day, right? And they were ready to stone him for it, if memory serves. So, and again, I'm punting to Walton again because he's the Hebrew scholar I know best.
He's coming. He's
Yeah,
coming July 20th.
Yeah, so you can press Walton when he's here this summer. I won't be here because I'll be in Oxford, but he'll be here in the summer at Regent. And is he coming here as well? Or
Yeah,
just
CSCA.
on the CSCA website, John Walton coming back. Walton's view of Sabbath is that it's not this, or the rest of God is not sort of the passive end of creation, but it's actually God taking up residence in the cosmic temple to actually begin running the show, as it were. That this is when a God rests in his temple, that's when he's taking up the reins of power. So this idea that all creation had to stop at the end of that rest, or at the end of that creation week, I would question whether or not that has a basis in the Hebrew understanding of what's going on there. I think it's probably time for people to